This is Natalia Elizalde. I'm Chief Business Officer at VIVEbiotech. So VIVEbiotech is a company fully specialized on lentiviral vectors. 10 years now as a company, so doing everything that is needed from very early stages to GMP, covering up to commercial phases. Specialized both in ex vivo lentiviral vector manufacturing and also in vivo lentiviral vector manufacturing, where we are having several programs, already 14 programs indeed, for different applications, such as in vivo CAR-Ts, vaccines, rare diseases applications, and so on. So pleased to have this discussion today, mainly focused on this in vivo. Yeah. What readiness means is that the vectors, the lentiviral vectors that are produced for in vivo, they should demonstrate a good degree of purity. So this is very key. Of course, they should also be cost effective, because cost of goods, they are, this is an essential topic for any kind of therapy, but very specifically, this is very, very significant for in vivo. So this is two topics: purity, cost effectiveness, of course, as well. And then the fact that the quality of the raw materials, they should be demonstrated to be the highest one from the very early clinical trial. So these three topics, they are very important to be ready for in vivo. The main bottleneck somehow could continue being the fact that the production processes, even though they are giving good results in our hands, for instance, but still they do need to be continuously optimized to have the greater titers, and therefore continue enhancing the cost effectiveness. Secondly, it's very important to continue working on different new pseudotypes that can target in more efficient manner the specific self to be reached, as well as other strategies, such as having, for instance, specific design vectors, decorating them so that they can evade the immune system, among other kind of strategies. So I think this would be the main topics. I think we are closer and closer, so the therapies that are being used so far in vivo, they are being demonstrated to be safe. The efficacy so far is being good, and also the cost effectiveness is being something to be highlighted. But still, as I mentioned at the beginning of the answer, there are some topics that should continue being improved and enhanced. And this is what we are really working as biology experts, having a dedicated department that can contribute to all these enhancements in the production. Regulatory is something very, very key for in vivo, and indeed having the regulatory strategy to be followed clear from the beginning of the development is, in this case, something essential as well. So, for instance, defining from the very early beginning the potency assay to be developed for this specific therapy is something important. So this potency assay should be validated even from early clinical trials. Also, the impurity profiles to be reached, characterizing very well the process, as well as all the IPCs all along the production process. This is also something very essential. So both the purity profile, the potency, the quality of the raw materials, and basically having the regulatory strategy clear from the beginning, and being able to have a direct interaction with regulators, letting them know what their plans are, so having the very best responses from the beginning, because this is a field that is now evolving, and I think that all the different players, we should work together towards this definition, also from the regulatory point of view. In my opinion, the ethical questions to be answered for in vivo use, they are the same ethical questions that for any other therapy development, not only advanced therapies, but any other therapy. So I do not see a specific difference from this point of view. It's clear that what is so important is defining very well the clinical trial design, having a good follow-up of these clinical trials, so pharmacovigilance in this case also plays a role, as it also does for other therapies. But at least as we see it, we do not foresee any additional ethical issue or aspect that should not be considered also for any other drug, any other therapy. So the technological breakthroughs that could have a major impact from our point of view, they are different pseudotypes. This is very key. Different kind of tissue-specific promoters, as I could already mention. Decorative vectors, so all that has to see with how the vector has been designed. This will make a difference itself. And also the fact that you should demonstrate not also high titers, but also reproducibility, batch to batch. And for that, different strategies can be followed. For instance, in our case, we are now also developing a stable cell line that could demonstrate this reproducibility from batch to batch, among others. So yeah, there are different lines that could cover all these aspects. From my point of view, this will be what the field will be doing in the coming few years. So AI is having an impact, from my point of view, in the sense that it makes the companies, for instance, the CNC process more efficient. It's a kind of a contributor. It helps. It's a tool. Of course, it's coming to help all the people working there, all the knowledge that does need to be put into such a complex kind of work. So I think that it will help to make the process more efficient and therefore diminishing the costs. This is something so important for in vivo therapies that from this point of view, this is something very positive. Indeed, for instance, at VIVEbiotech as a company, we do have a strategic project in which we are considering, together with a consultant, which are the best kind of programs we are running currently, in which AI could be applied, and always thinking on the efficiency of the processes, and of course on how this could contribute to our daily work as a team with such a knowledge in virology and adding value to it. So yeah, this is how I see.